not here. Okay, so this, usually when I do these notes, I do them as like art class, kind of. Usually I have everyone draw all of these parts, but I drew them for you just to keep it short today, and also to keep it easy with Zoom and all that. But this is the cell cycle. I draw it as a circle because that's what it is. It is constantly like happening over and over again in your body with your cells. Some of your cells don't do this, but most of them do. So some cells in your body will not ever divide, such as your nerve cells, which is why if someone gets like nerve damage, they usually can't really recover from that. Like paralysis is usually permanent. So that's an example of a cell that does not do the cell cycle, but for the most part, most cells do this over and over again, tons and tons of times throughout their lifetime and also throughout your lifetime. So we're going to go through the process. First of all, I want to tell you that the arrows are color coded this way for a reason. So all of the warm colored arrows, the pink, the orange, and the like peachy one are part of one like big chunk of the cell cycle, which is called interphase. So I'm going to label that on the inside of this arrow. And interphase includes all of these warm colored arrows. So it includes G1, S, and G2 all the way until the blue arrow. The blue arrow is mitosis, which is the actual like cell division moment. So this is mitosis, which is division. Put that in the wrong place. And the size of the arrows is also intentional. So mitosis is a lot shorter than interphase. Most cells will spend most of their time in interphase, which is just like preparing to divide and also just sort of like living their regular life. And we'll talk about that in just a moment. All right, so around the like donut of arrows, you will see a bunch of different drawings. Those are meant to be like the same cell going through the process. The blue is the DNA, and the gray is the membranes of the cell. So one is the cell membrane, and then one is the nuclear membrane or the nucleus. Let's start here with our growth one or gap one. It's called both, and you might see it called either one. And you also can just refer to it as plain G1. That's also perfectly fine. What happens during this time is exactly what it sounds like. So the cell performs normal function while growing larger. So the cell is just doing its regular normal thing, and also like eating food and getting bigger and growing in size. Throughout this process, we're also going to pay attention to what the DNA is doing. So in this situation, the DNA is in the nucleus and organized as chromatin. So that's a word from our DNA unit before Christmas, but the blue like spaghetti like structure inside the nucleus in my cell picture is the chromatin. And remember that you cannot see chromatin under a microscope. You would just see like a gray blob of the DNA. So I'm gonna say like not visible under microscope. And then if the DNA did organize into chromosomes here, how many chromosomes would it have for a human? You guys remember?
The 23 or like 46, right?
Yeah, it's 23 pairs, so 46.
Right.
You were right in both ways. There are 46 total chromosomes. So even though the DNA isn't organized into those chromosomes, they still exist. Like they're still there, they're just unraveled. So it's like saying, if you unraveled a bunch of thread and you were like, there's 46 spools of thread here, like there still is 46 spools, it's just not on the spools, if that makes sense. And that's G1. So it's also, I'll just say the longest phase. So some cells will spend almost their whole entire life just hanging out in G1, doing normal cell things, growing, eating, chilling, living the good life. For most cells, there needs to be some sort of trigger or chemical signal that tells them like, okay, now we're going to move on to the next step. Sometimes that's like an injury, and the immune system will release chemical signals that says, okay, time to make more cells to fill in this wound, or a hormone can trigger it, like growth hormone. There are different ways that cells can be triggered into the next step. Otherwise, they'll probably just stay here in growth 1, doing what they do. So you really don't want cells to divide when they're not supposed to. That equals cancer. We'll talk about that later. Okay, so let's say that our cell got, let's say our cell got a chemical signal. So we're going to go on to step 2, which is called synthesis. Synthesis basically means like produce things. In this case, it's producing DNA. So synthesis is the step of DNA replication. So DNA copies itself. So at the end of S phase, how many chromosomes will you have? 46 times 2 copies.
92.
Yeah, good job. 92 chromosomes. However, the chromosomes will still be organized as chromatin. And it will be in the nucleus. S phase happens pretty fast in cells. Before Christmas, when you guys made those DNA models, you did S phase, but you did it for only 6 base pairs. Your cells do it for billions of base pairs really fast, which is amazing. Think about how long it took you to just do 6. And your cells are doing it right now without any problems, which is great. So the only difference here between this cell and the first one is that the cell has 2 times the DNA, but it will look exactly the same as the first one did under a microscope. And that is S phase. The next step is G2 or growth 2 or gap 2. So after synthesis, the cell will immediately go into this phase called growth 2. I'm going to erase this what is cancer thing and put it back later just because we need a little more room to write. Okay, so in growth 2 or gap 2, the most important thing that happens is some checkpoints. So checkpoints look for mutations in the newly copied DNA. Mutations are errors. It's really important that the cell check for these mutations because if it doesn't, then the mutations will get passed on to the resulting cells. and they'll pass it on to their resulting cells and over and over and over again. So mutations can actually like build up over time because of S phase and checkpoints not working correctly. For the most part, your cells do a really, really good job of checking for mutations, noticing them, fixing them, or like aborting the mission and not dividing at all if they find a mutation. So I'm going to give the 3 options for what cells might do if mutation is found. Oops. There are 3 options. The first one is that the cell will fix the mutation. And continues with cell cycle. And a lot of times it's as easy as that. It's just like, okay, we fixed it and we're going to keep going. Especially if it's a really simple mutation, cells are able to resolve that really quickly using enzymes. The other options are less exciting. The first one is that the cell exits cell cycle and will never divide. When cells do that, it's called G0. The 0 is written as a subscript down below the G. So that basically means like it knows it has mutation, so it's not going to make any more copies of itself. It's just going to like live out its life alone. The last step or option is that the cell systematically kills itself. And that's called apoptosis, which is a fun word to say. But basically, if the cell realizes that like it is a danger to other cells because it has mutations that will cause it like harm other cells by replicating too quickly, it will like remove itself from the body through apoptosis. It's a way that your body can like safeguard itself from itself. And that's the 3 options of if a mutation is found. Does anyone have any questions about that? When I say systematically, it means like it will break down its organelles and then it will break down its cell membrane. It has like the enzymes it needs to like break down all the parts of the cell. Okay, cool. So let's say now that our cell like checked off the DNA is good to go. We're going to head on to the next step. The other thing that happens in growth too is a lot of growth. So this is much bigger cell. But same amount of DNA as before. Okay, so now it's time for actual mitosis. Here we go. You might have learned about mitosis before in middle school, so it might be a little bit of some review for you, but I don't know, all of you came from different middle schools, so it's hard for me to know. There are 4 steps of mitosis: prophase, metaphase, anaphase, telophase. You might have heard like PMAT before as a way to remember it. And then there's also a 5th step that's really fast that we'll get to in just a second. For each step, different things are happening in the cell, leading towards creating 2 exact new cells. So we're going to start with prophase. Prophase is the first step. You can remember this like prologue comes before, it's the first step. So usually pro means like before. That's what it means here too, because it's first. In prophase, a few things happen. The first one is that chromosomes form and become visible. So this is the first time that you would be able to actually see the chromosomes under the microscope. The other thing that happens is the nucleus actually breaks down. And this might feel strange to you, but it's really important because eventually we're going to have to get that DNA into 2 cells, which means it has to come out of the nucleus in order to achieve that. So the nucleus breaks itself down. And then also a new organelle that we haven't talked about before called centrioles move to opposite ends of the cell. These orange star-like things are called centrioles. And centrioles, interesting fact, are only found in animal cells. So plant cells do mitosis, but they do it a little bit differently, and we're not really going to talk about that. But animal cells do it this way using centrioles. And then, of course, I only drew 4 chromosomes, but in your cells, there would be 46. And the chromosomes are X-shaped now because they went through synthesis and they copied themselves. All right. After this happens, we can move on to the next step, which is metaphase. If you come up here it is. Metaphase is M for middle. Because what happens is the chromosomes make a single file line down the middle of the cell. Form single file line down middle of cell. And they attach to spindle fibers at their centromeres. So a couple vocab review words there. Spindle fibers are these orange, light orange, like spider webby things. They're made of proteins. They attach to the middle of the chromosome, and the middle of the chromosome is the centromere. So the blue dot, the dark blue dots are the centromeres. And then you can see in this picture, like there is no nucleus anymore because it broke down. And that's metaphase. So if they're all in the middle, it's metaphase. After metaphase comes the time to pull apart the chromosomes. So up here is anaphase. You can remember this one as A for away or apart. So what happens in this one is sister chromatids pull apart because spindle fibers shorten. So basically the centrioles like suck the spindle fibers back in, and that pulls apart the chromatids, which remember is 1/2 of the X-shaped chromosome, towards opposite sides. Now, one thing I do want to tell you is in this picture, it looks like the sister chromatids are V-shaped. They're actually lines, but they're being dragged through water by the middle. So think about that as like you're floating in a pool and your friend comes and grabs your waist and starts dragging you through the pool. And your arms and legs are just like floating. That's why they look like V's, but they're actually lines. And that's anaphase. During this time, also the cell is like stretching in both directions because it's about to divide. Now it's time for step 4, which is telophase. Telophase happens really, really fast. So fast that it's usually lumped together with step 5. But what happens here is that 2 new nuclei form around each set of chromosomes. So you have your new nuclei that are built up around your chromosomes. And then right after that happens, cytokinesis happens, which is number 5. And this means that the cytoplasm of the cell pinches together until 2 cells are formed. So it's like taking a ball of Play-Doh and pinching the middle until it breaks into 2 balls. This like dotted line here where the pinching is happening, it's almost like putting a belt on the, it's done by proteins. It's like putting a belt on the cell and tightening the belt until it like pinches all the way off. This structure has a name. It's called a cleavage furrow. That's the location where they actually pinch together. Now, in plants, can this work like this? What do you think?
No,
Yeah, why not, Jack?
because they have a cell wall.
Exactly. So I'm just going to put a little star over here that says in plants, there is no cleavage furrow. There is a cell plate that forms to split the cells. So you have basically like a rectangular plant cell and a cell plate like grows through the middle and it keeps getting longer and longer and longer until it cuts off the 2 cells, and then you have, that's your cytokinesis. This is cell 1 and this is cell 2 now. But again, we're not really focusing on plants. I just wanted to like mention that it's different. And then look where we are after that. We're right back to the beginning, to growth 1. So also during this time, the chromosomes uncoil. Back into chromatin. And then you have 2 choices. You can either restart the cycle, which is what this cell's doing, or the cell can actually exit the cell cycle, which is called, again, G0, which means it won't divide anymore. Just an interesting fact about this, your cells have little end caps on their DNA called telomeres. And every time you do S phase, you lose a little bit of your telomeres off the end. And when your telomeres get too short, that's a signal to your cell that says, we're not going to divide anymore. We're going to be done with this. And it contributes to aging. Because when your cells stop dividing, you're older. So your cells actually keep track of their age using their telomeres. Any questions so far? I just have a tiny bit more. All right, cool. So now I just want to look at this green blob I put over here. I wrote it for you already, but I just want to mention these things to you. So important things to remember about mitosis. First of all, interphase is much longer than mitosis. Mitosis only lasts about 24 hours in your cells, so it happens really fast. Interphase lasts really long time. Mitosis makes two identical daughter cells. The daughter cells are the cells that result. I don't know why they're called daughters and not sons, but they just are. Mitosis is considered to be asexual reproduction because you start with one cell and you make two. So you're making exact clone copies, and that's why it's considered to be asexual. And then I already mentioned this, but not every cell does mitosis, and some of them will never divide. Okay, just as a preview for tomorrow, the last thing I want to write is a little bit of stuff about cancer. So just come over to the side, and I'm going to say cancer overview. Is a cell that is dividing out of control. So it's making tons and tons of copies of itself. When it makes tons and tons of copies of itself, that is a tumor, a giant clump of cells that are dividing too fast. So I'm going to write here that cancer equals cells that are replicating out of control due to mutations. Usually it takes many mutations to get to the point where someone actually has full-blown cancer because, like I said before when we were talking about G2, your cells have a lot of ways to check for mistakes, fix the mistakes, remove the damaged cell, and your body's really, really good at that. So typically to have cancer, it takes a buildup of mutations that have just kind of snuck through over time, which is why for most cancers, they're more common in older people because those mutations have like built up. Every person who has cancer, their cancer is different from every other person who has cancer, even if they both have pancreatic cancer. The first person has different mutations that led to that than the second person does. So that's what makes cancer really, really hard to treat, is that the mutations that led to it are different. So usually you have people treated by chemotherapy. I'm going to write this, that chemotherapy is a treatment that targets and kills all rapidly dividing cells. So that's why if you know anyone who's ever had chemotherapy, you know that it is like a very painful experience and a really tough treatment because it doesn't only kill the tumor cells, it also kills every other cell that's doing a lot of cell division. And some parts of your body are supposed to do a lot of cell division, like your hair follicles and the lining of your throat and your stomach and your bone marrow. So people who undergo chemotherapy have a lot of side effects, which I'm sure that you know about. But that's the reason, is because there's not a better treatment right now that targets just the mutated genes in people. And that's why gene therapy is such an interesting discussion and an important discussion to have when we think about like the potential to treat cancer in a way that is better than chemotherapy or surgery or radiation. We're going to talk more about cancer tomorrow, but I just wanted to give you that little overview. Does anyone have any questions about it? All right, cool. Okay, so that is it for writing down notes. Everyone, you have another page in your OneNote below this. It's called Online Mitosis Lab. I'm going to just go over it really quick, and then I'm going to let you get off Zoom and do it. or stay on Zoom if you want to work with someone on Zoom. Okay. So first of all, I printed this to OneNote. So the link that's like right here is not going to work, but the link next to it does work. So click that and it will take you to this website. As you go through it, there are a few different questions for you to answer in your document. So actually read this because it will give you the answer. When you're ready, click next. It will take you to this picture of the different phases. This is a more realistic version than the one that I drew, obviously. In your document, it's going to ask you to do a basic sketch and then a brief description of what's happening. A way that you can already start to like study this is by trying to do this without looking at your notes. And then look at your notes and check your work and see if you're right. That's a way for you to like learn it better than just copying it off your notes. Try to describe it in ways that are like from your mind in a way that you understand it instead of just looking at the notes and copying exactly what I wrote. So once you do your sketches and your brief descriptions, come down here. You're going to see this chart on the website. Click next. The website is going to be like, draw this chart. I already drew it for you. So just click next. And then what I have for you is, or this has for you, is a picture of a cell and then the phases. The cell pictures are small. This little thing in the corner is the cell picture. You can zoom in your screen so that you can see it better if you want to. Then whatever you think it's in, you click that. So what is this one? What do you think? Where are the chromosomes?
Metaphase.
Good job, Jack. You're killing it today. I'm so proud of you. So once you know where you think it goes, you click where it goes. If you're right, your cell will go under that spot. If you're wrong, it will say try again, and you'll have to try again. At the end, you're going to have all your cells lined up in the sections of the chart. There's 36 you need to do. Once you've done that, count up how many are in each column, write the number here. The total is 36. Then you need to calculate percent. This formula down here tells you how to calculate percent. The only thing I want to tell you is it's saying the total number of cells is 36, not the total number of cells minus 36. It's just telling you like the total number of cells is 36. So use 36 on the bottom. And then once you do that, you just have some conclusions. And that's it. So if you would like to stay on Zoom, I'll open up some breakout rooms. If you would like to leave and work with someone else, like by FaceTiming them, or if you want to work by yourself, that's also fine. And I'll see you tomorrow at 9 a.m. Check your email for the Zoom link for that.
This is Darwin.
Yep.
And what is it
Chris, I can't hear you.
He said, when is it due?
Oh, Thursday.
Thank you.
Yep.